Hi everyone, in this video we will do a hands-on where we will explore how to use agent skills for single cell RNA sequencing data analysis. Okay, so basically we will use a skill document where we will be documenting all the single cell best practices, and now we will tell our agent to use the instruction given in the skill document to do the single cell data analysis for us. And again we'll be using Claude Desktop to show the demo. Okay, so yeah, so just let's recap like what we have talked so far. We see like with skills and without skills. Without skills, agent will use its own knowledge, so the results will be less accurate and less predictable. Whereas with skills, your agent will read the document and do the as per the best practices, so results will be more accurate and more predictable. And then we talked about different AI framework that is used like Anthropic, OpenAI, Gemini, and we'll be using Cloud Desktop for the analysis. And then we saw these two repository where life science related skills are already documented. And for this single cell data analysis use case, we will be using first because both of these repository has different skills. They have written different skills for single cell data analysis. First we'll start with this one, and then we will go with this one. Okay, and so yeah, and last video we talked, I gave a demonstration on like how to use NGS skills, and in this video we will focus on single cell data analysis. So without any delay, let's start. Okay, so if you go to this repository from Anthropic, life science repository, so they have already different skills available documented. So one of them, if you go, that's on the single cell RNA-seq. Okay, so here the main document is the skills.md file. I just open it into a new tab. So you can see that this is the main table of content. You can see this is what your agent sees. Oh, okay, the skill name is single cell RNA-seq, and what it does, it performs quality control. It performs quality control on single cell RNA-seq data either in this file using SCVerse best practices with med-based filtering and comprehensive visualization. Okay, so it also does QC analysis, etc. So this is what the agent will have the knowledge about, that hey, I have some skills for single cell data analysis. Okay, now when you ask an agent, hey, I have this data, can you do a QC analysis for me, single cell data analysis for me, then agent will go and actually read this content. So that means this much amount of information will not be loaded into token, so you will not be paying for that initially. Only when you ask, then only this will be loaded and you will be charged for the token. So that's what this is called, progressive disclosure. Okay, so now here it is written that what it does and when to use skills. For example, use these skills when a user requests QC analysis on any single cell RNA-seq data, and let's say they want to filter some low quality data and they need some QC related visualization, and they want to do some med based filtering like that, and the input currently formatted format is only this H5 format, and there are two approaches they have documented. So one is approach one is like complete QC pipeline. Basically, you take the file and you do end-to-end analysis. And for that, they have said like you should use this scripts within that. That's they have also provided that script over this folder. If you go to the scripts folder, there are three scripts that are available. QC analysis, this is the end-to-end, and these are the two sub. You can say that like if someone wants to do one component, they can use this Python script, and for plotting, they have another Python script like that. Okay, so that's what they have written over here. If you want to use complete QC pipeline, you can directly follow this one. This is how they should run it. Python 3, this one, and you can give the input file. Okay, and this script automatically detects the file format and loads it appropriately. And when to use this approach, so this is standard QC with adjustable thresholds, and batch processing, use this one. Quick exploratory analysis, you can use this approach one like that. Okay, and what are the requirement to, we need to install these packages and data scanpy, all these Python packages is needed, and this is what parameters that this command needs. And also user can use this med count, empty threshold. So while interacting with agent, you can also specify that hey, I want to minimum cell should be this much, a med count should be this much, that much you can tell about the agent. And output will be saved into this kind of file you will get it. And yeah, so what the workflow does, this end to end, it does the QC metrics, then apply the MAD based filtering. MAD is like median based filtering, and it will do the filtering genes and also generate the visualization. This is the approach one. Okay, so now the approach 2 would be modular. So for example, it is also says like so for modular you can use this second function qc core, or let's say qc plotting.py. So you can see, right, you can also, when you are creating your own skills, you can modularize your scripts in a different format. Okay, so that no need to run end to end. You can also interact with the agent to do step by step. Okay, run step 1, like QC, just give me a QC plot, then next do the filtering, next do this one. So like that you can break, and interactively, iteratively you can with your agent you can work. So that's the beauty of organizing your code in a different files. Okay, and also you can give some example. Let's say just example I'll show you. So it says like when to use this approach. The second approach, like modular approach, let's say different workflow needed. For example, you can skip some steps, or you can change the order, or maybe you can want to apply some different threshold, and or let's say some conditional logic, like for example, filter a neuron cell different than other cells, those kind of thing. Or let's say partial execution, that means you want only filtering, not downstream analysis, like that. And it says that what are the different functions available in that script, and what each function does. From this QC plotting, there are 3 functions are there, and what each function does. And some example also it has given. For example, if a user asks only calculate the metrics, don't filter yet, then this is what you should follow. You are telling this agent, you are training your agent, you can say. When your agent reads this one, it will understand how it will do that. For example, a user can ask like apply only mitochondrial percentage filtering, whereas keep other metrics permissive. So then you can use this kind of method. Or you can try with different threshold for different subset. Then you can follow these approaches. And then you can also given some best practices. You're telling that, hey, default threshold intentionally retain most cells to avoid losing the error population. So that's the best practices. Okay, go with the default one. Always review before and after plot to ensure filtering makes biological sense. So that's what you are giving specific instruction to the agent, where it can follow the best practices to give you the answer. So this, without skills, your agent will randomly will hallucinate, and it can give any random answer. But with skills, it will try to adhere the best practices that you have strictly asked it to. Okay, so furthermore, you also given that for detailed QC methodology, parameter SNLA, and troubleshooting guidance, please refer this one. For example, user can ask, what is MAD? I don't know, like why I should use 5%, why not 8%? Then agent should be able to answer, right? So that's where for further detail, agent will be now go and read this one, and then based on that it will try to give you some answer. So very beautiful, like, so means like you can organize your skills like very the way you organize books, right, chapter wise, and for the reference kind of, like for the Wikipedia first page, you click the link, you go to the second, so like that for the reading kind of thing, you can create such a nice skill structure. So that's what progressively the agent will disclose each information, and it will read it, not like everything dumped. Okay, so then further suggestion, like usually after this one should do this kind of thing, but this is not possible because that is not provided in this folder. But you get an idea, right? This folder, what you have, it has the main skill.markdown where everything is documented. It has the scripts, and it has the further reference material. Okay, so you can download this and then you can zip it and you load it with the cloud to work. So that's what we will be doing. Okay, and I already have cloned this repository over here, and so if I go to this single cell RNA QC, I just have to zip it. Okay, so let's create a zip file, compress to zip file, and yeah, so my zip file is ready, and I just copy this path, and I go over here, go to Claude Desktop. This will look like this, and you can go to files and settings, and if you go to capabilities, and in the past video I have added 2 QC, but now let's add one more QC skills, one more skills. Upload this zip file and give this path and load this zip file. Okay, so now it will load the single cell skills. Okay, that's very interesting, and you can click it just to see that what the agent will see. The agent will see only this description, it will not see, but when you ask it, then it will able to see, it will read everything. Okay, so now let's open a new chart. First ask better to navigate, hey, what local agent skills you have. Okay. So bioinformatics, now you see there are three skills I have added, right? So single cell RNA sequencing skills are there. Okay. So now let me add some files there. Um, already, so this is already I have one PBMC data is there. So I will ask him like, can you do, let's do end to end. Okay. Let's see what it does. Can you do single cell data analysis of this file? Also tell me what steps you followed and what skills you referred. Okay. Okay, so like that you should ask like okay, once I, so that I will also show like like how the second approach will go. Okay, so let it go. So now you see, once I ask, it now reading the single cell RNA-seq skills. Okay, so it already read it. So now it is further checking some scripts. So you saw that okay, it has all the skills available, these three scripts are available. So now it needs the required packages, because in the skills we have given right what all packages is needed. Again, once again I'm telling that this is where it is running. It is not running on my local system, it is running on the Anthropic server. So because I have the account, right? So in Anthropic server somewhere, for my user account, a sandbox will be created, and within that, this session, basically whatever this chart interface is going on, a container will be created, and that's where everything is running. So, and all the file what we uploaded will gets uploaded there, and all the execution happen, we will get the output. But once I close this chat window, everything will gone. So that's why we have to download it. So alternate approach is like, this is for demo purpose and you can just trial it out. And if you want for long-term actual use case, then you should switch to cloud code or cloud SDK agent, those kind of thing, but those are paid one. Okay, so yeah, let's see, it installed it I think, and now it is running the command. And see, it is copied these three skills over here, and it is now running the QC analysis pipeline. Let's see what it did. So yeah, so it is now running the single cell analysis and it is storing all the results over here, and these are the parameters it used, and it loaded the data, and see, voila. So you have all the nice very amazing. So you see how it did. So it says the like, so now I have asked her what you did, right? So it said telling me I use the single cell RNA-seq skills located at this location. This location is in the Anthropic server, which follows the SC-verse best practices with MAD based filtering. Specifically, I ran the complete pipeline via this QC analysis.py, which is the approach one from the skills. See, there are two approach I explained, right? So now what it steps followed, it first loaded the data, and this many cells, this many genes were there. Then it calculated the QC metrics, and these are the QC metrics it calculated. And it also says like how many mitochondrial genes it found, how many ribosomal reads, and how many hemoglobin reads it found. Then it did some pre-filtering QC plot. So plot 1 it says, so this is our plot 1. This is QC metrics before filtering, all those images you will see. And then it did yeah, then it did the MAD based filtering, and then MAD is nothing but median absolute deviation based threshold, which is more robust than standard deviation based. And then after filtering, what happened, total 92 cells got flagged with respect to this QC matrix, and around 89 cells got flagged with respect to this bad quality with respect to genes, and with respect to mitochondria percentage, around 200 cells got flagged, and 8 additional cells removed from the hard empty threshold. So like that, total how many, 312 cells removed. And after filter, it again tried to do the QC analysis, and finally gave me the output like this. So next step is recommended approach is double-edged, these things are there, but just very important, like this is not part of skills, because my skills what we uploaded, right, this does not contain anything. So but it is now using its own knowledge. But if you ask like, can you do double detection here, it will use its own knowledge to do this analysis. That's where you can add one more skills, for example, for double detection, maybe they follow the same SC DVL finder, and then you tell that use this one to do the doublet detection. So then it will be more accurate than completely relying on this agent. We don't know, right, how good bioinformatician this agent is all about. Okay, so that's what, again, but this gives you a quick idea. So now I want to try with approach 2. Okay, of these skills. Okay, just load the data and tell me what to do next. Let's do step by step. Okay, so this is how agent, I want to try the second approach. So now it's already installed, right? So now first step is like it will load the data, inspect it. So it followed the same instruction that we have given, and so data loaded. Here it is. So like it is there, and total genes like that. What's the next step? Step to calculate QC matrix. So for that it will be using this function. You see, it will not use the entire pipeline, and this function is there in this QC matrix, right? Let's check that. Approach 2. We are seeing that for calculate QC matrix, right? It will be using this one. Okay, so let's do step let's do step 2. Okay, so now it will exactly do the calculate QC matrix, and it will give me an output. So now it is now running the script. See which script it is trying to find. So it use that calculate QC matrix command, and it is now generating the plots for me, and for that it use the plot distribution function, and now you will get, I think, minimum similar output you will get. But here it gives additional information like total count, how many was there, what was the median value, what is the maximum value. So that's the advantage of if you do the end-to-end analysis, you will get final output. Okay, but if you go step by step, it will try to explain you at every step how it went. So purely annotation, no filtering yet. So now let's say run step to calculate matrix. Okay, yeah, go with filtering, for example, filter. So again here we can further ask, let it complete. We will ask like what threshold it used, and we can ask again, can you do with this threshold? Let's see. So it is now running, run full filtering using approach to modular function. It completed, and okay, so it gave me some output here, and you see at 5x mad 92 cell removed, like that. Okay, so I can ask you like what cutoff or okay thresholds were used for filtering. Okay, so total MAD 5. So because median was this one, MAD 499, flag 92 cell. Okay, so can you use MAD 8 for let's say total read depth, and redo. Okay, so let's see it is doing or not. So now this time it should use yes, it used 8, right? So for total counts, let's see. So that's the advantage of just don't give entire script like complete pipeline. No, always give, when you're writing scripts like building, break down into different component kind of thing. Basically write different functions and let the agent call the function automatically. Okay, so now you see MAD 5 total 92 cells were there, but after MAD made 8 total counts, it got 21 cells. So that way, so it says that wider MAD8 window kept around 13 more cells, mainly the ones with higher read count that were previously flagged as potential doubling. So it also gives you the knowledge about, right? So that's the beauty of doing this kind of analysis. Okay, so now so that's what I showed you how you can do that from using this entropic this life science skills. But alternate, as I said, the this bio skills is there at GPT Omics. They have also given this skills document. Yeah, they have also given the single cell analysis. If you can go single cell, right? but they have even more comprehensive. So basically you can do a lot of things like batch correction, cell annotation, cell communication, clustering, data input output, doublet detection, lot of things. So let's do one thing. I will also show you this example. We can go with data IO. Okay, so basically here this will help us to load the data, and the main thing, like, it also supports both Surate and ScanPy. Okay, the previous one was using ScanPy, so let's this time try to go with Surate one. I will show you how to do that. We'll use, because this tells like you can do with Surate, and it also give the code for how to do in ScanPy, and also how to do the same using Surate. Okay, for example, you can load the library, you load the data, and then object structure is like this: create a matrix, read this one, add the metadata, all these things, everything it does. This is like data IO. So once we load this one skills, and then let's do one thing. I'm just thinking about pre-processing, right, is another skill. So we load this one. So here what it does, like, so here we can do all quality control, filtering, normalization, these three we can do using Surate. So here, so first we will be loading you. I'll ask the agent to use these skills to load the data and give some summary, and then use this pre-processing skills and to do the pre-processing, and then probably we will ask him to do something called clustering. So we'll see the clustering skills, and that's where we will use, like, PCA and Surate and ScanPy. And fourth skills what we will be using is, let's say, called cell annotation. So these are the four steps basically in any pipeline we follow, right? So I think it will be able to do that. Okay, let me, I have tried it, just a second. We go after clustering. I think marker annotation probably. Let's see. Find marker annotate the cell types in single cell RNA-seq. Use the differential expression between clusters. Okay, so let's do this one, marker annotation. Okay, so what we will do, okay, so I've already cloned this one. So let's go to this, the bio skills. Okay, and then we will go to single cell there, right? Okay, so now let's do one by one. First we will do the data.io. Okay, and here, as I said, you open this one, and it has two additional line, uh, which, it will give problem in Cloud Desktop. So I deleted that and saved this one. So this is my first data.io, and I will just zip it. Okay, so now once data loaded, then we will do the processing, right, pre-processing. So again, open this one, just edit this one. Because this other extra line what I'm deleting, right, those are not recognizable by the Cloud Desktop, so that's why it is giving the error. Okay, so zip it. So we have two skills ready. Now once we do the pre-processing, we'll go with the clustering, right? So yeah, let's open this one. So I just delete that, these two. So clustering skills is ready. I will just zip it. And the last one what we will do is, let's try with the marker annotation. Okay, let's open this one, close this one, zip it again. Sorry, I have to do it. I'm doing it. uh, but for the safe side, I will also do, why to take, we did the marker annotation. What will do, cell annotation, let's do that. Okay, that also, in case that doesn't work, I think. Okay, let's see. Okay, AG method. All right, so now zip it. Okay, perfect. Ready? So let's go. Okay, let's close this one and go to files and settings. Okay, and go to capabilities. So I have already these three skills are there. I will just, currently I'll not use this one, just disabling it. And now let's add one by one skills, and let's just copy this path so that I can. Okay, so first load the data.io. Now I will add preprocessing. Problem is like I cannot select all at one go. I think it is not allowing me. So clustering. So now let's upload one more skill that's on about the marker annotation. Okay. And last one more skill I will upload, that's the cell annotation. Okay. So see, I have added 5 skills, but only those table of content, the front tool and name and description, that is only will be known to your agent. Okay. So now let's chart again. Hey, what local skills you have? So copy this path. So now see, it has now the name got changed because bio cell data IO 5 skills it has. Okay. So now let's to upload this file. Okay, go back. This is my PBMC data. Can you load this data? Okay, this single cell data. Okay. So now what it will do, it will now see it automatically. It will use the data IO skills because that's where we are loading it. And since it's the first time it is doing, it's a new chart interface. It has to install the scanpy and end data. Okay, so let it install. So it installed. So now it is running the command. It is loading the data, and yes, so it loaded the data and it is giving me some information. What's next? I can run the standard pipeline on this. Yeah, run. Yeah, pre-process this data. Use skills. Okay, I'm just explicitly telling it to use skills. So now it is now reading the pre-processing skills. Good, I have the skills. Now let me run the full pre-processing pipeline. So now it is running the script and the instruction, whatever it is there, and it is doing some filtering and it is storing the raw data. It is now normalizing and log transforming. It is calculating the highly variable genes. Okay, okay. So it is doing it. Okay. So now let's do one thing because it used scanpy, right? So I wanted to show you, Surat, right? So let me stop this one. Hey, can you use Surat instead to load the data and pre-process? Okay, because already I have shown it will behave the same. So let's try the Surat one. So since it now need R, right? It will install R and all the dependencies. It took some time to install actually. Lot of things it did to install, but finally, it did pre-processing pipeline. Okay. So I had asked like, can you install load the data and pre-process, and so just check. Did you use skills for this, and what skills? Let's just for testing. Yes. So yep. So it's saying that correctly. Yes, I did use skills for this. Specifically, I read 2 skills. For one is the data IO to load the data, and then I read this pre-processing to this one. Okay, so the output you see, right? This is what the Surat plot will look like. You see while in scatter plot, you will see, you know, and highly variable genes plot. Okay, and yeah. So how many variable genes you used? Okay, so I think the default was 2000, right? So that was used. Okay. Okay, so now we have done the processing. Next, what can you do? Okay, so we can go with clustering. Okay, so let's go with clustering. Okay, so now it will now read the clustering skills. Okay, got the skills running the clustering pipeline. So basically, so what does the clustering skill says? Let's see. So it will do this kind of command like run PCA, find neighbors, run UMAP, and it will do the different parameters, reference value, low, high, all this information is given here. So let's see what it does now. So it is again the same thing. It is loaded. It is done the PCA, run PCUC, and yeah, it is doing loading plots, finding neighbors, finding clusters, and UMAP it is doing. Okay, very interesting. See, UMAP coloring, and it is giving the output. So let's see. We very soon we will get the output. I'm excited. So now you see progressively how we are each skill. So we have 5 skills loaded. Not everything is done one time. So first data input skills, then we are telling the do the pre-processing one, then you can do the QC skills, then do the clustering like that. So let's see. So it has copied the output. Now you get 9 clusters identified at resolution 5. So these are the Louvain clusters, the UMAP, total 9 clusters is found. And it also tells like each clusters how many cells are there, how many pieces were used, and cluster size ranges from this many to this one. Okay, so plots generated is like that. So this is the first plot. The UMAP resolution, you can see at different resolution how the clustering almost similar, only 877. And these are the top markers with respect to that, how so for example the CD3D is belonging to this cluster, like that. And the elbow plot, PCA loadings, and which genes correlated with which PCA, like that. Okay, so next what we do marker marker identification. Okay, let's see. So now it will do the that skills, marker annotation skills. It is reading now. So here basically we are explicitly telling like we can use this to library and find all markers between different cell clusters. Also it will use this marker detection method, and then it will do some plots, and I think it will use particularly these genes maybe to annotate, because these genes are mainly for T cell score. And let's see what it does. And it will try to manually annotate it. And if these markers are present, then these are T cells, B cells marker, like that. Okay, these are some examples it has given in the skills, but you can further guide our agent like how to interpret it. So let's see what it is running now. So yeah, it is doing this find all markers. I'm sorry for that. Then top 5 clusters, then top 10 clusters heat map, then it is doing the heat map. It is doing dot plot with these markers. Amazing. See, these are 8 clusters, and how these genes important for the 0, 1, 2, 3, 4 each clusters. And these are the top markers it detected, it used. So these are the dot plot, like how for each genes and which clusters how they are expressed and all. Same for the top markers, the violin plot, their expression in which clusters. And yeah, top 5 markers also you can get in CSV file as well. Okay, so this is the a differential expression. This is how the zero does, and you are able to do it. So yeah, last can you do cell, what is the last step? What can I do next? Okay, I don't remember, so I'll ask him. So let's see. So you can do the cell type annotation. Okay, let's do this one. So last, this is where I will stop, but that will give you can get an idea. I don't know, I've never tried this whether it will work or not. Let's see. So they're using single R for this, and I think it is not installed probably. Okay, unable to locate package. It will try to install an alternate approach, and it may take some time. So it is doing something here. Is checking install or not. Yeah, it is installed. Okay, cell disk first failed, so it will now use bioconductor to install, and some network block is there, I think. So yeah, so that's why it's saying cell dx is not available due to network restriction, because this is running in their server, right? Something it will do. Okay, So then it will try to use its own knowledge to do something. Okay, so it did something. Okay, this it will do that one. Okay, okay, let me wait to do that. Let's see what it is doing. It is doing generating some plots. All the outputs are done. Now running the command, it is generating the output, right? I think it is already done. Yeah, it generated some nice output like B cell, T cell, like that. It generated UMAP cluster versus cell type, and then dot plot annotated cell composition, which cell contains percentage of cells, feature plot key markers. Amazing, amazing, amazing. So you see, right, in short, like I try to give a demo on like how we can use skills very easy. Like if you're expert in a given field, you just document in a skills.md and provide extra scripts and all, and just upload it and let the AI do everything for you. So this is how the future of all analysis will look like. But as I said, AI can't replace because it is just doing what you have given. That's where your domain knowledge will come. So yeah, I think that's all for this demo. Next, I will talk about, like, I can just show you one quick example like how you can write your own skills in the next video. I hope this video was useful, and if yes, so thank you for watching. I will see you in the next video. Bye bye.